I welcome you all in the SWAM course on genetic engineering and its recent advancement. Today I am going to speak on the cloning and expression vectors, and as we know the gene cloning is a major breakthrough and we have to understand what is the role of cloning and expression vectors. Before we are going to start with the in detail about the cloning and expression vectors, let us understand what is the role of vector in gene cloning. So, we know that these are the important tools. We understand first there are many tools which are important to conduct the series of experiments in the genetic engineering and biotechnology. So, here we will see what is the role of vectors and what are the two types of vectors which are very important tools for the genetic engineering and biotechnology. So, here we will see the very important thing, what are vectors. Vector is kind of a DNA molecule. It can be said as a fraction of DNA that is used to carry or transfer the genetic material from one organism to another organism. So, we see here the vector as a vehicle molecule for the transferring of DNA molecule from one particular cell to another type of cell. And as we know, the first important thing is to express or to clone any DNA molecule, we need to first create the recombinant DNA molecule, your genetic engineering. So, recombinant DNA, the first constructed recombinant DNA molecule is the vector itself. So, it is a fraction of DNA which carries the gene of interest into it, and it acts as a vehicle for transport of the new DNA molecule from a novel source to a kind of host organism, where it is used to create multiple copies or numerous copies of it. So, this is a very commonly used tool in the genetic engineering, and we know there are two types. So, based on the functions you can clone, you can propagate, you can reproduce, as well as you can manipulate and express the gene of the interest, because gene is a segment of the DNA sequence which carries the important information. Information, when you see the final product, is always the protein, and we say that this is the expressed form of the gene. So, gene of interest has to be expressed in regarding to the kind of application where exactly we are doing the genetic engineering. So, the first important thing is to transfer the DNA molecule, to make numerous copies of it, and once you are able to manipulate it, then you can do the various changes in that particular gene of interest. And with this new addition or new modification, you are also able to take the expression or the formation of the final product into different type of host organism. So, the host is the where we actually produce the numerous copies, which is exactly called as the cloning. Cloning is making the numerous identical copy of the DNA molecule. So, here we see there are the first factor is the cloning vector, and cloning is the important technology, and we know by literature it means that cloning is to make the identical copies. So, vector role is here in the cloning vector is to produce the or you can say amplify, that is the number of the copies of the new DNA molecules which is carried by the vector DNA is amplified, and when the host cell divides, then it is going to be propagated into the all the daughter cells. So, this is how we see that cloned DNA fragments. So, the very first step in the DNA cloning mechanism is to create the recombinant, and as we know the DNA, all DNA, they share their chemical structures, they have their same kind of chemical structures, but they differ in their sequences. So, if they have the same chemical structure, it is very much possible to create the recombinant DNA. It means at least two different strains are required to create the recombinant DNA. So, the vector DNA is a fraction of DNA that takes the interest, means your insert or the gene of interest or the DNA of interest, and the first recombinant DNA is the vector itself. And then we see to do all this kind of activity, because we have to carry the DNA, you have to make the numerous copies of it, and then it has to be if it is a cloning vector, then it has to be propagated. So, in regarding this we find that there are certain important properties which makes the vector or a carrier molecule or a DNA molecule as a cloning vector. And for this, what is required? For the cloning, there are certain unique sites should be available that makes the process convenient, and we can use it as a tool for cloning the DNA molecule. So, there are some features, the major features which we include as a common or standard DNA sequences are selectable marker, the presence of selectable marker, multiple cloning sites and origin of replication. So, here we will see that in each of this component which is present at a DNA sequence in the cloning vector are equally important, and they are essential sequences to be present in any vector to act as a vector or cloning vector. So, one by one we will see that what is the role of selectable marker. So, this is the kind of DNA sequence which carries the some of the important traits within that, and which allows us, right, for the identification and the selection of cells containing the vector. So, this is a very big question which comes in our mind, that once you transfer the vector to the host cell, we have to confirm whether it contains the recombinant vector or not. To understand that, practically what we have to do, we have to have some detectable sequences, DNA sequences, which makes us easy to identify and select host cells, because it is a transfer from one to another. So, that the host organism, whether it contains the vector DNA, we need the marker sequences, and it makes ease to identify whether the given host cell is having the vectors or the recombinant vectors or not. And very good examples are like these selectable marker, mostly they are the certain type of antibiotic resistance. So, what will happen if the vector is having this selectable marker, suppose for ampicillin resistance? So, only the vector containing host cell will survive in any media, select your screening media where the media is supplemented with the antibiotic. So, non vector containing cells will die, and only the vector containing host organism will be surviving, and that gives this property comes because it contains the antibiotic resistance marker gene which is available in the vector molecule. So, selection is a very important phenomena, and detection is a very important phenomena. Once you successfully get the clones, you have to find out whether it is a successful event has happened or not. To find out that this probability, this selectable markers helps the experimenter to identify the host cells containing the vector or the recombinant DNA. So, remember one more thing, the vector is the DNA which is the first recombinant DNA, because it is carrying the DNA which is not its indigenous part, it is non-native to the fraction of DNA. New DNA has been inserted to create the vector, and now the vector is moving, right, and you have various tools by which you can transfer the vector to the host organism. The second important sequence is the multiple cloning site. So, this is again a unique site, it is a fragment of the DNA sequence where we find out it is a location. We simply see cloning site means what? It is the location which is present in the vector where we actually insert the DNA fragment of your choice, that is the what DNA fragment you want to clone, you want to make the amplified copies or the numerous copies, there is a specific site in the vector DNA fraction which allows to uptake the new DNA of your interest. And the reason behind it, why it is able to take, because it is the unique site for certain type of restriction enzymes. So, it is a recognition sequence for the restriction enzyme. Restrictions enzyme, when you add in the in vitro system, there itself the enzyme will act, it will open the DNA molecule so that the new DNA can be inserted. And good thing is that you have multiple cloning site, it means not a single site recognition sequence for the restriction enzyme. However, there are many types of restriction enzyme recognition sequences are present, and as per your choice you can apply various type of enzyme in your in vitro mixture of the cloning experiments, and this one you apply the enzyme, enzyme will recognize the site, it will cleave that site, it will open the site, and then one can easily insert the DNA. So, this is how the very first step, the recombinant DNA has been created in vitro condition. So, you have one DNA of vector and one DNA which is novel DNA, new DNA or the DNA fragment of your choice, and then once the recognition sequence is present, one can easily open the DNA molecule and you can insert this DNA to the vector DNA, and vector DNA is now ready to take the DNA from one cell to another cell. Coming to the next type of vectors. So, there are common features which are shared by the cloning and expression vector, but the major function of the cloning vector is only to carry the DNA, to transfer the DNA, right, that is the essential feature, and to make the numerous copies. So, that is all about the cloning vector, but there are multiple fates may exist for this new DNA. If you are designing the vector in such a way that you carry all the necessary regulatory sequences around the DNA of interest, which allows the expression of this cloned DNA. Expression means it is now you have all the efficient, or we can say the several essential features, right, which is present, which allows this cloned DNA to be expressed. This is called to be the gene expression. So, you have a gene, you are successfully cloned. Now, you do not want only to clone and to make the numerous copies. Your interest is different here, you want to express it. Express means you want that new gene which has arrived to the host cell should also make the final functional protein. And if you want that protein to be expressed, then obviously as a part of the gene expression system, the cloned fragment should carry all the essential feature which allows the efficient expression of this cloned gene. So, now you have to see that what are the regulatory sequences which is needed. Obviously, cloning and expression, if together you want, then you need the transcription and translation. So, these are the various, we can say that the two important steps of the gene expression, and to transcribe the gene you need the important gene segment which regulate the transcription, like you need the promoter sequences, transcription initiation sequences, then you have to terminate the transcription for a proper control over its transcription, and similarly once you want the expression in form of translation. So, once the mRNA is formed, the next step is the how to translate that expressed mRNA, and to start that you have to add the ribosomal binding site, that is the transcription translation initiation codon should be there, stop codon should be there. So, these are all the regulatory sequence which is required for the transcription and translation. So, the cloning vectors you add new features. So, we can say the cloning vector can be modified to make it expression vector by means of adding certain important necessary sequences which regulate the expression of the gene in terms of transcription and translation. So, unique more such sites are created which helps the experimenter. So, this is a kind of created vectors, right, and you are adding some regulatory sequences. So, the gene which is cloned, now it is ready to start the transcription as well as the translation. So, you will see that promoter will be there. Promoter is a very important site where the RNA polymerase it binds and then it initiates the transcription, and then the ribosomal binding site is the first part of the this mRNA transcript to start the efficient translation, because the mRNA is there, ribosome will bind over there, and downstream to this ribosomal binding site there will be a translation initiation or the start codon will be there, and this mRNA transcript is without any like interruption because of the presence of the translationary regulatory sequence, it is also going to be translated like a natural phenomena of gene expression. So, that we are making those features we are creating in the form of expression vectors. So, this is how the expression vectors are also made in gene cloning experiment, where your interest is to make the final product out of the gene, or the vector is transporting it, it is taking the new DNA, but at the same time the new DNA or the gene sequences or the clone genes in the host organism is also expressed. So, as I told you, the cloning vectors are the DNA molecules. So, their major function of the cloning vectors are to replicate and propagate the cloned DNA fragments. So, this is how we summarize, or we in one line, if you want to understand about the cloning vector, it is taking the recombinant DNA and making numerous copies of this DNA into the host organism by using the host cell machinery of replication. So, the only function is the replication. The new DNA is replicated. If it is replicated, means it is amplified, and when the host cell divides, it also then transferred to the daughter cells, and how there is a propagation. So, you can get a DNA fragment, multiple copies of it, which is actually the cloning process. So, this is a very essential tool in the genetic engineering, and this is how we can do the cloning. And then you have also choice to change the DNA or the gene of interest, and further you can use it for the various type of basic and applied studies. So, I am going to talk in the detail about this, all the features, important features or the DNA sequences which is necessary to be present in the vector to act as a cloning vector. The very important thing is the origin of replication. So, you know, what I am telling here is that it is going to replicate. So, we want the site in this fraction of DNA which is acting as a cloning vector should contain the site of origin of replication. So, this is independent to the host cell genome. It is not at all depend on the host cell genome, because all this replication is going on inside the host cell, but the replication of the vector is independent to the host cell genome. It means that it does not depend on the host genome, even it means it can divide, it can replicate even though the. So, what happened in the origin of replication? What is the importance of origin of replication here? That this is the site which gives the freedom to the vector to independently replicate within the host cell, and this is essential for the amplification of the cloned DNA fragments. So, host cell genome is also having the origin of replication, and the vector is also the DNA molecule which itself carries its own origin of replication. So, it is not depending on the host cell genome, it can divide, it can multiply, it can replicate, right. It can replicate basically independent to the host cell genome, because it is having its own origin of replication, and this is required to make the numerous copies of the cloned DNA fragments or for its amplification, right. So, this is what actually is the cloning. So, we can amplify the number of copies of the DNA fragments just because it is having its own origin of replication. So, this is one of the essential feature to be present in the cloning vector. Then you see the multiple cloning site, it also contains numerous multiple cloning site, which is also called to be the polylinker site. Why it is polylinker site? Because it is a kind of short DNA sequences, and these DNA sequences actually act as a, or this contains the recognition sites, they act as a recognition sequence for several unique restriction enzymes. So, this recognition sites allows to be identified by the restriction enzyme, and when the restriction enzyme recognize the sites, it will cleave on that, and that will open the vector DNA molecule, so that the new DNA can be inserted into the vector DNA, and this is how we can even say it is a linking site. That is why polylinker site, because multiple choices of restriction enzymes are present in these sites, and one can apply depending on the restriction enzyme and their property different type of restriction enzyme to digest the cloning vector for the introduction of the gene of interest. And the third part is the selectable marker. So, the one more important sequence or a DNA sequence which has to be present in the cloning vector is the selectable marker, right. So, this selectable marker is the property of the cloning vector, which is present only in the vector genome, not in the host cell genome. So, this is a specific phenotype, specific traits given, right. It is giving the specific property to the cell by, say for example, here would be like a antibiotic resistance. So, there maybe ampicillin or tetracycline resistance is there. So, the cell are resistant to this treatment, antibiotic treatment, because of the presence of the resistant property of the vector given to the host cell. So, if the cloning vectors has successfully reached or introduced to find out the successful recombination has done or not, to identify or to select the cells, right, containing the vector, this marker helps us. That is why it is called as a selectable marker. So, when you grow the host cell, once the vector is transferred, then when what you do, you grow the host cell and you treat in the cell, your screening media you provide with the antibiotic, say for example ampicillin. So, this kind of cells which carries the vector will be able to survive in the antibiotic containing media. Why? Because of the presence of the resistant property due to the cloning vector. So, this is helping for the selection of the cloned host cell, and that is why they are called as selectable marker. Size is another factor, right. So, size is like cloning vectors, normally we see they are small sized and they are circular DNA molecules, that is like few thousand, 100 to few thousand base pair sizes present. So, they are mostly the small circular DNA molecule, and this type of thing means the vector DNA or this type of DNA fractions are naturally present also in various type of microorganism, and you can also design, right, a new type of vector as per the requirement also. So, mostly they are small circular DNA molecule, and compatibility means these vectors when you design, you find out that it should grow, it should be able to be transferred into different type of host organism. Why? Because for the replication it requires the host cell machinery. Independently it cannot divide without cell. So, it has to be, first you do the recombination, you create it, and then you insert or introduce the vector into the host. So, host is the indispensable type of tool. You need that host where whatever the host you are selecting, the vector should divide, right, the vector should grow, the vector should multiply inside that, and that is why we find out ki which type of microorganisms can be used as the host organism. So, that varies depending on the vectors, but we find out the compatibility that it should grow in bacteria, yeast and certain type of other mammalian or eukaryotic cells also, because if we are creating or you are designing in the lab, we definitely are interested that such vectors are able to grow in multiple type of microbial cell, so that you can transfer the cloned DNA fragments into different type of host organism. Particularly it is desirable if you are working with the like for different type of DNA fragments of the sizes of the DNA fragments also. So, here we see ki as I know this is what DNA fraction can be said as the cloning factors, what are the essential features of it, and now we search in the nature whether such type of carriers are already present. Yes, they are there, and very good examples are the DNA molecule which can act as a vehicle for the transfer are plasmid and bacteriophages. So, they are very well present in the nature. Nature, you know that many of the bacteria, right, they have the plasmid, even some yeast cells they have the plasmid, they are short DNA molecules, they can be used as the vehicle molecule. Phages, bacteriophages are the second one. They are naturally the transferring viruses because they are intracellular obligate organisms which are the bacterial eater virus. So, this is bacteriophages are the viruses. Viruses always do their replication inside the bacterial cell. So, this is the natural phenomena the phages is having. They are highly efficient to introduce their DNA into the bacterial cell, and there inside the bacterial cell they divide, means they divide, and before dividing the whole replication of the phage DNA is occurring inside the bacterial cytoplasm. So, they are present in the cytoplasm, plasmids are there in the cell cytoplasm, phages are there in the cytoplasm. Now, the next type of the vector is the expression vectors, and these are specially designed DNA molecules for the expression of the gene of interest as I told you. So, let us see the various features first of the expression vectors. First is the, like it is having various important necessary regulatory elements to be present, like transcriptional initiators, right, like promoter sequences, enhancer sequences, and at the end of the gene segment you will see the transcriptional ending sequences, terminator sequences. So, this is overall to regulate the gene expression and for its efficient translation. So, the coding sequence will be there. So, as you know, when you talk about the gene expression, there are the features of the transcription, there are many segments, regulatory sequence of the translation is there, and everything to be present in the designed vector as a expression vector to complete the expression, right. So, we want the efficient transcription and translation, and to do that, to achieve that, all the regulatory sequences of the transcription like promoters, transcriptional initiation, terminators for the translation like ribosomal binding site, start codon, stop codon has to be present to get the product like protein. So, protein, and you know the expression vectors, they have a various applications because you can express in form of protein and you can use it for the various applications, like you can make peptides, various type of proteins, right. Your pharmaceutically important proteins and peptides can be created. So, this is like expression vectors are specially designed vectors to achieve the gene expression. So, these are the same sequences which has been shown in the PGX 1, 2, 3 series, and you can see this having all the important sequences. You have the gene of interest, and this gene of interest is flanked by the all the important sequences to be acting as the. So, along with your origin of replication, selectable marker, polycloning sites, this is also essential, which is the commonly shared by the cloning. Apart from that, the other regulatory sequences like transcriptional start sequences, stop sequences, promoter sequences, ribosomal binding site sequences like signed Dalgarno sequences are also present, which is flanked by the gene of interest for its expression. So, this one which I have just now told, that expression vector will have the promoters, right, for its to start the transcription. It is having the multiple cloning site where you exactly insert the gene of interest, and then you have the transcriptional initiator promoter where the RNA polymerase binds, and then the transcriptional terminates is the signal which is present at the end of the gene of interest to stop the or to terminate the transcriptional process, and then you have the regulatory sequences which can also increase the level of gene expression. This is another kind of sequences to be present. You have origin of replication, right, this is always important. So, it is shared by the cloning vectors, like it is also having the independent origin of replication for its replication or for its amplifications are there. More than that, you will add more signals like polyadenylation signals are there, right, for the it acts as a regulatory sequences, and this is how the expression vectors are used, right. So, it is carrying all the regulatory sequences. So, once it reach to the once it is introduced to the host cell along with the your vector is having the foreign gene. So, this foreign gene is expressed here. So, this is a difference between the cloning and expression vector. It is not only making the numerous copies, you will see that mRNA and protein is formed in case of expression vectors within the host cell. These are the major differentiating points between the cloning and expression vector. So, their application is different. First is only to make the numerous copies, whereas to express the gene, this expression vector will allow you to express the gene of your interest also, right. The insert size may vary, right, and the promoters or the certain type of sequences, right. It is not all the regulatory sequences which is required to express the gene are absent in the cloning, whereas it is all present in the expression vectors. And selectable marker is the essential feature for the cloning, but it may or it may contain, because you select the host cell based on the formation of the protein, functional protein. So, this is all about like major distinct point between the cloning and expression vector. So, going to the end, I will say that there are two basic type of vector, that is the cloning and expression vector. They share many of the features, but we can say expression vector is the more advanced version, because it is the major objective to create or to design the expression vector is not only to make the numerous copies of the gene of interest, but also to express in the form of final product. Why? Because you have designed it, you have added all the regulatory necessary features into it, so that the gene which has been cloned can also be expressed. Thank you once again to all for this topic of course.